Hello, welcome to biology class. I am Shipra from ADARSH MANDIR SHANKAR for class 12. We are starting new chapter, molecular basis of inheritance. In this chapter, we will study about the nucleic acids. Nucleic acids are the building blocks of genetic material. So, what are the genetic material? Genetic material is the substance which not only controls the information and expression of the traits, but also it can replicate and transfer the information from parents to the offsprings. And in our body, two types of nucleic acids are present, which are RNA and DNA. RNA means ribose nucleic acid and DNA deoxyribose nucleic acid. DNA is present in the nucleus in the form of chromosomes, chromatic material, and RNA is present in the nucleolus. Nucleolus is the spherical solid structure which is present inside the nucleus. So RNA is helpful in the protein synthesis and DNA is a genetic material in our body. So what are the basic features of the DNA? DNA is a polymer of deoxyribonucleotides. So first, what is nucleotides? Nucleotide is the unit of DNA which is made up of mainly three components: nitrogenous bases, deoxyribose sugar, and phosphate group. Phosphate is present in the form of phosphoric acid (H3PO4). Deoxyribose sugar present in DNA, and at the place of deoxyribose sugar in RNA, ribose sugar is present. And nitrogenous bases. There are two types of nitrogenous bases present in DNA and RNA called purines and pyrimidines. Purines are the double ring structure, and adenine and guanine, two types of purines are present in DNA, and pyrimidines, cytosine and thymine, two types of pyrimidines are also present in the DNA. But at the place of thymine in the RNA, uracil is present. So what are the differences between the RNA and DNA? RNA is single stranded, while in comparison to DNA is double stranded. At the place of thymine is present in DNA, and in RNA uracil is present. Uracil is slightly different from the thymine, and it is a 3-methyl. Thymine is uracil, and at the place of deoxyribose sugar in the RNA, ribose sugar is present, and in both the RNA and DNA, phosphoric acid is present as a phosphate group. So we come to the nucleoside. What are nucleoside? Nucleoside is a combination of one nitrogenous base with pentose sugar. Pentose means 5 carbon sugar. They are bonded by the N-glycosidic bond. Clear? So what are the combination? The adenosine. What is adenosine? Adenosine is the combination of adenine with ribose sugar. Ribose is a pentose sugar. So this is known as adenosine. It is a type of nucleoside. Guanosine. Guanosine is a combination of guanine with ribose sugar. Cytidine. Cytosine with ribose sugar. And uridine. It's very important. Uridine means uracil with ribose sugar. It is present inside the RNA. It is a nucleoside of RNA. And what about the nucleosides of DNA? They are using deoxyribose sugar. So they are known as deoxyadenosine. Deoxyadenosine means adenine with pentose sugar but deoxyribose sugar. And deoxycytidine means cytosine jointed with the deoxyribose sugar. Like this. This is the nucleoside. Now what are the nucleotides? Nucleotide is a combination of one nitrogenous base plus one molecule of pentose sugar and one molecule of phosphoric acid. So this combination is known as nucleotide, and all the nucleotides are jointed by the glycosidic bond and phosphodiester bond. This is the phosphate, and phosphate is connected with two nucleotides. This is one nucleotide, one nitrogenous base, one sugar and one phosphate. This is other nucleotide, nitrogenous base, sugar and phosphate, but these are connected. by 2 times. This is known as phosphodiester bond. Clear? And what is the structure of DNA? The double helical structure, the double helical model of DNA is given by Watson and Crick. And what is in the structure of model? These two strands are anti-parallel to each other, and the backbone of the molecule, this is the backbone, is made up of sugar and phosphate, and the stairs of the, these are the stairs of the, we can say the stack, they are made up of nitrogenous base pairs. These are the nitrogenous base pair. So this makes us double helical structure, and this joint, both the strands, this is the one strand and this is other strand, both the strands are jointed by the hydrogen bonds, and the combination of hydrogen bond is fixed. Always adenine is connected to the thymine with the help of 2 hydrogen bonds. Guanine connected with cytosine with 3 hydrogen bonds. Clear? So these are the hydrogen bonds. They are very weak bonds. They can easily break, and the difference between the 2 pairs, these 2 nitrogenous pair, the distance is fixed. The diameter is 20 angstrom, and the turn, one turn, this is a one turn contain 10 base pairs. One turn contain 10 base pairs. Like this is the, these are 4 base pairs and 10 base pairs, and the length of one turn is 34 angstrom. So the difference between the one base pair to the another base pair is 3.4 angstrom, because the 10 base pair have 34 angstrom difference. So the one base pair, it is 3.4 angstrom distance. Clear? So this is the structure of DNA. And what about RNA? RNA is a single stranded. So the nucleotides are jointed by diester bond, but only one strand is present. Now the next is central dogma. What is central dogma? Central dogma is given by Francis Crick, and he gives the information about the flow of molecular information. So first give from DNA to RNA and RNA to protein. The synthesis of DNA to DNA is known as replication, and from DNA to messenger RNA is known as transcription, and messenger RNA to protein is known as translation. So here is the 3 reactions. What is? First is replication. Replication means formation of DNA to DNA. Transcription means from DNA to messenger RNA. But one more is the reverse transcription. What is reverse transcription? Formation of RNA to DNA. This is known as reverse transcription. So this is all about the DNA. Thank you.